Good morning, 10Hodgkin. Welcome back. Hopefully you had a enjoyable and restful half-term break and were able to enjoy this lovely weather, to get outside for some of the time. I hope you are looking forward to your final half-term in Year 10. This short video is just going to focus on the feedback for the assessments that you did just before half-term. I'm going to look specifically at the biology and the physics ones. Miss Bailey will be feeding back to you separately about the chemistry. Okay, so the first point is around your individual feedback. So on Microsoft Teams you will see that there is a new assignment, okay, which is simply the dirt work, the feedback for these assessments. There's one for each of them. On those I will simply be returning, you don't need to send anything in just yet, I will be returning those to you with your feedback. Okay, that will include your individual score and some key areas for development. Okay, these are based on specifically the areas that maybe you struggle with a little bit more in any of the tests. Okay, they will involve some work on Seneca. Just to recap, go back over and study a few things. Okay, will be the main tasks. If you have any questions about that, please do get in touch. Okay, so let's focus on some of the whole class feedback for biology first of all. So a couple of misconceptions and things that I picked out that were just general points, okay, that a lot of people sort of stumbled on, so I'm just going to go through those first of all. First thing is about the respiration. There was a question about respiration. And it's worth remembering that all living things respire, and that includes obviously bacteria. Okay, a lot of people left bacteria out of that question, but bacteria obviously do carry out respiration. Second point that I picked up was a very common one, which was getting during the experiment, the photosynthesis experiment, which talked about the difference between repeatability and reproducibility. Okay, so that's something that we do need to kind of understand, and we'll go into a bit more detail when we are back in class. Okay, but essentially what we mean by if an experiment is repeatable, it means that we can there is no variation within the measurements. Okay, so if we can repeat something, okay, it means that you can carry out that, and you get constantly, you get very little variation across the measurements that you take. If something is reproducible, it's simply that means that that result can be repeated, it can be reproduced. Okay, and that would be the same for perhaps the whole experiment. Another question that I think a few people stumbled on was the key things that plants need to survive, okay, being water, minerals, space and sunlight. So they obviously don't compete for other things other than those four key points. And then the other main thing that I think caught a few people out was really just the wording around the term metabolism. Okay, so what we mean by metabolism is the sum of the chemical reactions that take place in living organisms. Okay, so it's all of those different things that happen throughout your body. Okay, the sum of those together is what we call metabolism. Okay, now then, the dirt tasks, which as we're going through now, as you can see, I will post this slide also onto the team site, so you have this available, but you have some consolidation tasks. So the first things are your individual feedback, which I've just mentioned already. Okay, they are specific Seneca tasks, ideally, for you to go back now. Some of you did some Seneca revision, okay, so this may be going back and just recap it, just embedding some of that. Some of you may not have used Seneca previously for the revision, so it would be worth going over and looking at those. Okay, if you feel that you've done as much Seneca as you can, okay, and you want to try something else, you can always go and use Educate. You can go and use the Tassomai that we've set up, okay, or any other things that you think would be useful. But I would suggest you start with the Seneca as a recap here. The second thing, okay, is one of three tasks. So just from memory only, okay, this is a kind of what do I know. I want you to pick one of these three things to just create a mind map with as much information as you can. Okay, you may want to do all three. So the different organelles and their functions that are found in animal, plant and bacterial cells. Okay, so 3 separate sheets, or however you want to lay it out, all of the things that you can remember. The structure and the functions of the features of a leaf. Okay, so what it's and also the rest of the plant even, or the carbon cycle. So I'm going to pick one of those for the moment. Okay, try and choose one that you are going to have to think about a little bit more. The develop task is again something that is individual to you. Okay, some people will have the same question here, but it is an exam question that I've set that kind of builds on what we've been doing. So it's related to the stuff we're doing. Okay, the stuff that was in the assessment, but it builds on that. So it's another type of question. So I'd like you to have a go at doing that. Okay, and that will be submitted back in. And then your stretch task, okay, so if you feel that you've done all of that and you really want to push yourself a bit further, is to have a go at writing your own exam question and a mark scheme. So pick one of the topics that were covered in the assessment, have a go at writing an exam question, ideally not a 1 mark answer, ideally look for something that's 3, 4, even 6 marks, okay. Remember to include the command words that'll help people, prompt people to try and answer it. And then also obviously the number of marks that are available. Now for submitting all of that stuff, okay, it will go, when I've returned the assignment to you, you should then be able to add stuff on and then send it back, submit it back into me. Okay, and that will be due at the end of this week, also Monday next week, the 8th of June. So that is the biology. Okay. The following slide is basically the same but for physics. Okay, so I'm just going to very quickly allow you to stop now. You might want to pause. You might want to come back to this part of the video when you look at your physics at a later point. Okay, or you might want to skip to here. So, going through the physics. Okay, 3 main things that I picked out. Okay, were the differences between the types of resistor. Okay, so fixed resistors, variable resistors, thermistors, LDRs, those sort of things. Something for you to kind of really focus on there, and there's some stuff in the consolidation task, as you can see. The V equals IR equation, where a lot of people kind of got mixed up a little bit with that, okay, and obviously rearranged it slightly incorrectly. Okay, so if you're trying to find resistance, you need to remember how to rearrange that equation, and it's a division of I over V rather than I times V. Okay, and then the IV characteristics graphs that looked at the diode, the film bulb, and the LDR, I think it was. I forget off the top of my head. But anyway, the IV characteristics graphs. Please go back and check your notes for those differences to try and make sure you understand which one is which. Okay. So again, dirt tasks, as mentioned. Same thing, individual feedback on your Teams site for the specific Seneca tasks to complete. I'd like you to, looking at those resistors, I suggest you create some flashcards for those different types. So what they are on one side, the symbol on one side, and what it is on the other side. Make sure you've got that. If you've not, if you've got those already, then go back and have a look at them again. And then I'd like you to use the link, which is one of the free science lessons videos, to help you attempt or reword your answer to question 8. Okay, so a lot of people kind of struggled with that question. Okay, not only in this class, across the board, struggled with the wording of it or how to go about doing it. So I'd like you to try and watch that video and then have another go at answering it. Okay, again, there's an exam question as a developed task. And then if you get onto it, a stretched question as well. Okay, I'd like everybody to have a go at the consolidation tasks as a minimum for both this and the biology. Again, any questions, please do ask. I'd love to know how you're getting on. I will be checking in with you obviously during the week. We have a Q&A planned. Okay, so if you have questions for me for that, please do bring them along or send them to me beforehand and we can answer them during it. I will dedicate some specific time during that Q&A at the start to answer any questions that you've submitted and to go through some of this feedback in a bit more detail. Okay, that's all for now. Have a lovely week, and I will speak to you later on this week.